Hello friends, welcome back to my YouTube channel, The Zone of Anatomy, and today we are going to discuss one of the most complicated, or we say the most complicated topic we take. So the topic for today is carbohydrate metabolism, and this series we are going to complete in one shot, which means we are going to cover all the cycles in the carbohydrate metabolism, and we have already discussed glucagon and insulin metabolism, that is carbohydrate metabolism. In our series of diabetic ketoacidosis, you can watch that series, and the link for the same is put in the description box. So go and watch after seeing this video or before seeing this video. So let's proceed it. We are going to discuss the cycles in this. So first of all, we need to learn this complete table, which means what is anabolic pathway concerned with carbohydrate metabolism, catabolic pathway, amphibolic pathway. Coming to what is anabolic pathway, anabolic means the metabolic pathway that construct molecules from the smaller units, like a very small unit combined to form a larger unit. So in this anabolic pathway, we have glucogenesis, that is synthesis of glycogen from glucose. So it is glycogenesis, that is synthesis of glycogen from glucose. Then we have a gluconeogenesis, that is formation of glucose from non-carbohydrate substances. This is the most favorite MCQ coming in maximum examination. Like they give the MCQ, synthesis of glucose from non-carbohydrate substance is known as, and the options are glycogenesis, gluconeogenesis, and glycolysis. So the option will be gluconeogenesis. So this one is one of the most important part which you need to learn. Second is lipogenesis. Lipogenesis is the synthesis of fatty acid and fat. Then we have a uronic acid synthesis. So this is our anabolic pathway. And what is anabolic pathway? It is the construction of the big molecule from smaller molecules. Coming to the catabolic pathway is basically concerned with the breakdown of big molecule into smaller molecules. Like for example, we go for glycolysis, that is oxidation of glucose. We are going to convert glucose into pyruvate and lactate. Pyruvate and lactate we see when we produce pyruvate and when we produce lactate. Lactate in anaerobic condition of glycolysis and pyruvate in aerobic condition. Another one is GLYCOGENOLYSIS. So breakdown of glycogen to glucose or glucose 1-phosphate. Then we have an HMP shunt. Another name for HMP shunt is pentose phosphate pathway, or oxidation of glucose to ribose sugar and reduced NADH. Another is oxidation of pyruvate to acetyl-CoA, that connecting link between glycolysis and TCA, that is tricarboxylic acid cycle. Coming to amphibolic pathway, then amphibolic pathway consists both anabolic and catabolic. So this is the only concern with TCA cycle, which is the metabolic pathway where acetyl-CoA is oxidized. So TCA cycle, another name is for citric acid cycle or Krebs cycle, and we know it is the common metabolic pathway where our acetyl-CoA is oxidized. So when we make a note that in the liver, muscles, adipose tissue, and the brain are the main site of our glucose metabolism, let's see what are the fates of the glucose metabolism. Coming to the fate of glucose metabolism, first is that glucose or the oxidation through glycolysis and TCA cycle and HMP shunt pathway. Second is storage as glycogen in liver and skeletal muscles by the process known as glycogenesis, Then conversion of fatty acid and storage in adipose tissue, that is glycogenesis. We see this all when we study about the protein metabolism or the amino acid metabolism time. Then conversion of non-essential amino acids to by the process known as amino acid synthesis. Then we see like non-essential amino acid, we see how our fatty acids, glyceraldehyde is converted to the glucose. Then we see synthesis of structural material. What are structural materials? Structural material can be glycoprotein, glycolipids, mucopolysaccharides. These all are our structural materials. Then we have a conversion of glucose into glucuronic acid, ascorbic acid, and pentose by our pathway, that is uronic acid pathway. But make sure that uronic acid pathway is not in the humans due to the reason that is absence of one enzyme, which we see when we talk about this pathway in brief. Then we need to remember one more thing, that the product of aerobic glycolysis, that is our pyruvate, when enters into the mitochondria for further oxidation in TCA cycle. Then coming to this glycolysis, what is this glycolysis is concerned about? First, we need to know what is the synonyms of it. Synonym means what is its another name? Another name is Amden-Meyerhof pathway. It is one of the most important. Then we see what is the glycolysis. Glycolysis is basically the oxidation of glucose or glycogen into pyruvate. Pyruvate in the condition known as aerobic and lactate in the condition of anaerobic with the production of ATP. The complete process is known as glycolysis. When we talk about two processes, that is aerobic and anaerobic, aerobic is the presence of oxygen and anaerobic is the absence of oxygen, and the product is pyruvate and it is the lactate. Coming to the sites, the product which is formed, that is pyruvate, it is in the all the cells which contain mitochondria, and the lactate where cells do not contain mitochondria like the RBCs, and the cells which have a few mitochondria like WBC, renal medulla, brain, GIT, retina, skin. This is one of the most important MCQ which might come about the aerobic condition and anaerobic condition sites. And the site is cells and tissue, as we see. Then we come to the compartment. What are the compartment? That the glycolysis takes place in the compartment known as cytoplasm. Its nature is catabolic. We have already seen in our first slide. Then we come its unique feature. Unique feature is obviously that it can be happen in aerobic and anaerobic condition, and its product can be pyruvate and lactate. Then how it is regulated? It is regulated by the stimulation of insulin. When we see insulin metabolism, we studied this about. Then there is the this glycolysis pathway is stimulated by insulin, ADP, adenophosphate, and AMP. Then we have inhibited the glycolysis inhibited by glucagon, that is glucagon plus ATP and citrate. One more thing you need to remember, in stimulation we have ADP, while in inhibition we have ATP. Do not make mistake in exam. Examiner might confuse you between ADP and ATP. ATP is concerned with glucagon and insulin. ADP is concerned with insulin. Coming to the total ATP production. So the total ATP production in this is 8 ATP in the aerobic condition, but when we open our Harpers, we might see at a 7 ATP also, and 2 ATPs from anaerobic conditions. Now, we have already know what is the glycolysis. You can open your textbook, whatever textbook you are preferring. So in that you can go through our complete glycolysis pathway. There, just go through the complete glycolysis pathway or just read it once, so you get an idea what is aerobic and what is anaerobic condition and everything. When we differentiate between aerobic and anaerobic is similarly in aerobic condition, there is the breakdown of glucose in the presence of oxygen, and in anaerobic, there is absence of oxygen. So the end product is pyruvate in aerobic, and end product of anaerobic is lactate. Another difference which we can is that The ATP production in aerobic is 8 and in lactate is 2. And the size, it is cells containing mitochondria, and the sites containing, like these are the complete differences which you need to know while attempting the question. Then we see what or how the glycolysis produce the ATP from 1 mole of glucose. First, we see in the aerobic condition. When we talk about the aerobic condition, we see the substrate level. We see what are substrate level? The substrate level is the reaction. What reaction? 1,3-bis, when we go from 1,3-bis phosphoglycerate to 3-phosphoglycerate, and the enzyme used is phosphoglycerate kinase. So that is a substrate level phosphorylation, and the ATP produces 4 ATP produces. When we go to respiratory chain oxidation, then the 6 ATP produce. So the total ATP is 10 ATP. You might come to know how this 6 ATP is produced, because we have 2 NADH2 and 1 NADH2 contain 3 ATP. So we have 2 moles of air. So we have 6 ATP production. So the total ATP produces 10 ATP, but we utilize 2 ATP. When we utilize 2 ATP, where we utilize this 2 ATP? In the step first, that is from glucose to glucose 6-phosphate, and in third step, that is fructose 6-phosphate to fructose 1,6-bisphosphate. There we consume our 2 ATP. So the net total production in the glycolysis is 6 ATP when we concern about the aerobic condition. So aerobic condition produce our 6 ATP, and the utilization is 2 ATP. 2 MCQs from this box. Then in anaerobic condition, when we talk about substrate level, we have a 4 ATP similar to this. Then the utilization of energy is 2 ATP. So the total ATP produces 2 ATP. So in anaerobic condition, we have 2 ATP production. This is the only thing which we need to remember by heart. So as I say, consumption of energy is from glucose to glucose 6-phosphate, and from fructose 6-phosphate to fructose 1,6-bisphosphate. So this you need to remember completely. Now, moving forward, what are the importance, or we can say, if you will, what are the importance of the glycolysis or EM pathway? First, glycolysis is the only pathway that occurs in all cells and all tissues. Second point, it is both the principal route for glucose metabolism, plus we say it is the main pathway for metabolism of fructose, galactose, and all other carbohydrate products. Then glycolysis is the only source for energy of RBCs. Why RBC? Because RBCs do not have a mitochondria, so it undergoes anaerobic condition for the production of energy. Then moving forward, when the person is hypoxic state, that means there is the lack of oxygen, then our heart muscles get energy by the process known as aerobic glycolysis. Then glycolysis is the only pathway which has both anaerobic and aerobic condition when we exercise. So glycolysis is the major source of energy for the muscles, since at that time there is a production of lactic acid. So we need an aerobic glycolysis. Then during, it is the main step for oxidation of glucose also. It is, or we say it is the preliminary step of complex oxidation of glucose. Then moving forward, when we talk about the metabolic pathways that occurs in the RBCs, we say RBCs has anaerobic glycolysis, that is without, it do not have mitochondria and no glycogen. Then we have HMP shunt, that is only source of NADH in RBCs. Then the important point which we need to know about the RBCs is that in RBCs, since we have mentioned that it is the aerobic condition, so there is no respiratory chain. Respiratory chain means ETC, electron transport chain. There is the no citric acid cycle or TCA cycle. So there is no oxidation of fatty acids. Similarly, RBC is highly dependent on the glucose as a source of its energy. So do not forget about this. Then when we talk about how important is this glycolysis in RBC, so just remember one point, that is RBCs are completely, completely reliant on the source which is known as the glucose as their metabolic fuel. Metabolic fuel and metabolize it by aerobic glycolysis. Second point which we need to remember is that glycolysis in RBCs produces 2,3-bis phosphoglycerate, which binds to our hemoglobin that decrease its affinity for oxygen, and so making oxygen more readily available to tissues. That's why we use the pathway known as the glycolysis. So in the glycolysis, you need to know, when we talk about what is glycogen hemoglobin oxygen curve, we come to know. But till now, you need to know that in RBCs, the anaerobic glycolysis plays an important role. When we talk about the inhibitors of glycolysis, we say the inhibitors are enolase. Then we have the inhibitor that is known as a fluoride. Fluoride inhibit enzyme enolase, and these two inhibitors we need to learn. And another one is iodoacetate. Here you see there is iodoacetate and arsenate, which inhibits the enzyme known as glyceraldehyde 3-phosphate dehydrogenase. If you go through the cycle, you can easily recall what are these like enzymes. When we talk about in vivo, glycolysis is inhibited by glucagon. When the blood plasma glucagon is high and insulin is low, we can see this in the diabetes. That's why I'm saying just go and read the diabetes lecture so you can easily come to know what is this. When we talk about the rate limiting enzyme, so the rate limiting enzyme is fructose 1,6-bisphosphate, and the enzyme used is phosphofructokinase with the utilization of ATP. One ATP is used in this. Coming to the irreversible step, there are 3 irreversible steps in the glycolysis, and you need to memorize by its heart. It might come, that is first step, that is glucose to glucose 6-phosphate, and the enzyme which is used is glucokinase and hexokinase. Where is glucokinase? Glucokinase is present in liver and pancreatic B cells, and hexokinase is present in extra liver tissues. Okay, so this you need to learn. Another point you need to learn about glucokinase is that it has low affinity for glucose, whereas hexokinase is high affinity for glucose. So glucokinase is not inhibited by glucose 6-phosphate, whereas hexokinase is inhibited by glucose 6-phosphate. Write this down as an MCQ, it might come. Then it is glucokinase is affected and stimulated by insulin, whereas hexokinase is not affected by insulin. So these points you need to remember while studying these two enzymes. Another one is fructose 6-phosphate to fructose 1,6-bisphosphate, and the enzyme uses phosphofructokinase. And last is our phosphoenol pyruvate converted to pyruvate, and the enzyme uses pyruvate kinase, and this is one of the most important 3 steps which are irreversible reactions and the major site of regulation of glycolysis. So you need to know this. Then the disease concerned with, we already studied about the enzymes. So if we talk about hemolytic anemia is concerned with the deficiency of enzyme known as pyruvate kinase. Fatigue is due to phosphofructokinase, and lactic acidosis is due to pyruvate dehydrogenase. Just remember these disease with the deficiency of their enzymes. Moving forward, when we talk about oxidation of pyruvate to acetyl-CoA, so let's understand important facts about pyruvate. Then we see important points about acetyl-CoA. So we have already discussed complete about glycolysis, then coming to the oxidation of pyruvate to acetyl CoA. So what is the meaning of conversion of pyruvate to acetyl CoA? That is, pyruvate dehydrogenase complex which we form is irreversible and it converts into pyruvate, and then pyruvate is converted into the acetyl CoA. Its silent features, or we say its substrate, is pyruvate. Its product is acetyl CoA. Its site is tissues and cells. Its compartment is mitochondria. Its nature is catabolic. That from small molecules it become a big molecule. Then its rate limiting enzyme is pyruvate dehydrogenase. ATP production is 6 ATP. That means 3 ATP for each pyruvate, and we have 2 pyruvate molecules. Therefore, the total ATP produced is 6. But according to Harper's, it is 5 ATP. So just make sure you go through it. And its unique feature or its significance is that it's a central step for the connection or the central step for linking glycolysis with TCA cycle. When we come to its fate, what are the fate of pyruvate? So the fate of pyruvate is that it has acetyl CoA, lactate, alanine. So fate of pyruvate. Fate of pyruvate is basically concerned about pyruvate is converted into acetyl CoA by pyruvate dehydrogenase complex, then lactate by lactate dehydrogenase under aerobic condition. Just write it down. Lactate in anaerobic condition by enzyme known as lactate dehydrogenase. Then alanine by transamination. When we talk about the process of transamination, then in the amino like when we talk about in the transamination, we see what is this alanine, then we say glucose by gluconeogenesis, then oxaloacetate by carboxylation, and ethanol by reduction in microorganism. So these are the fate of pyruvate. Guys, if you haven't learned or go through any cycle till now and your exam is very close, just read this. These are the only MCQs which your examiner might ask. These are the just a short notes which you need to remember, and it is actually very helpful. So if you haven't studied anything, don't be panicked. Just go through these points and make sure you write all the MCQs which are narrating, so it makes easy for you to differentiate or to attempt in the exam. And whatever points is mentioned in the slide, just remember by heart. Moving forward, enzymes in the pyruvate dehydrogenase complex. So enzymes are pyruvate dehydrogenase, dihydrolipoyl transacetylase, and dihydrolipoyl dehydrogenase. These are the 3 enzymes which you need to remember. Then we come to coenzymes in pyruvate dehydrogenase complex. What are the coenzymes which we are using over here? So it is thymine pyrophosphate, that is TPP, then lipoic acid, then coenzyme A, NADH, and FADH. So how the question come? Question come like, name the enzyme and coenzyme in pyruvate dehydrogenase complex. So you need to know the enzymes and all the coenzymes over here. Then we come to the sources of pyruvate or the pyruvic acid, and it is very easy. That is glucose oxidation by the process glycolysis, then lactic acid oxidation, then alanine deamination, and the catabolism of some glucogenic amino acid, that is glycine, that is serine, etc. We see when we talk about amino acid metabolism. Till now you need to know catabolism, that again I'm saying that is from small molecules you are making big molecule. Then another part we need to discuss is about acetyl CoA. So in acetyl CoA, what we say, acetyl CoA is a 2-carbon compound produced when our coenzyme A reacts with acetic acid. So about acetyl-CoA, the points which you need to remember is that the active form of fatty acid is acetyl-CoA. Write it down, it's an MCQ. Free fatty acid is converted into acetyl-CoA in a reaction which is catalyzed by acetyl-CoA synthetase. And the enzyme is thiokinase. And here the utilization of 1 ATP is utilization of 1 ATP is present. So the complete reaction which you can write is fatty acid plus ATP plus coenzyme A in the presence of enzyme known as acetyl-CoA synthetase produce our acetyl-CoA plus inorganic phosphate and AMP. This we need to remember the basic thing about acetyl-CoA. Then its sources are very easy: oxidation of glucose via glycolysis, oxidation of fatty acid by beta oxidation, oxidation of amino acid, degradation of ketone bodies, and oxidation of ethanol. Sources you need to know about acetyl-CoA. Then what are the fates of coenzyme A? So fate is oxidation in TCA cycle to produce carbon dioxide, water and ATP, synthesis of fatty acid in fed state when the person is eating, synthesis of cholesterol, synthesis of ketone bodies in starvation when the person is not eating, then formation of our acetylcholine. How acetylcholine is formed, we have already seen in yesterday's lecture when we talk about the neurotransmitters. So when there is the donation of acetyl group for synthesis of acetylcholine, like acetyl-CoA we have, it combines with the choline and it forms our acetylcholine. So remember this thing that it comes in the fate of acetyl-CoA. So this is our TCA cycle. So what are things we need to remember about TCA cycle? So first go back. What we know about TCA cycle, that it is a series of reaction taking place in mitochondria that oxidize acetyl-CoA and reduced coenzymes that are reoxidized through our electron transport chain linked to the formation of ATP. So this thing we need to know, or in the simple way we can say that it is one of the most common metabolic pathway that oxidize our acetyl-CoA to reduce our coenzyme A. What coenzyme A we are reducing? That is NADH, FADH. So this we need to remember. We are reducing coenzyme A, and it is the most common metabolic pathway that oxidizes our acetyl-CoA to reduce our coenzyme A. This we need to remember. Moving forward, TCA cycle is also known as Krebs cycle and tricarboxylic acid cycle. Its substrate, as we know, it is acetyl-CoA. Its products are reduced coenzyme A. What are reduced coenzymes? Reduced coenzymes are FADH2, NADH2, CO2, H2O, ATP. Its site is cells which containing mitochondria. Compartment is mitochondria. Its nature is amphibolic. Why its nature is amphibolic? It has both anabolic and catabolic part in itself. So we say that the TCA is amphibolic. When the examiner asks you, how can you distinguish that the TCA has both, like catabolic role also and anabolic role also? When we concerned about the catabolic role, we say that the catabolic role, just write it down this point, concerned with the TCA cycle is that TCA cycle is clearly catabolic because here our acetyl-CoA is oxidized to the product, oxidized to produce our ATP and carbon dioxide. And when we talk about anabolic role, we say because we call it as anabolic pathway because it produces various intermediates for the synthesis of many compounds. For example, what compounds it is synthesis? It synthesis are fatty acid, cholesterol, steroids by acetyl-CoA. It also synthesis are non-essential amino acid by oxaloacetate and alpha-ketoglutarate. Another one, succinyl-CoA is also used for the synthesis of heme. Then it is intermediate of TCA cycle. Then it produces purine and pyrimidines. So these are the anabolic pathway which is concerned with TCA, that's why we called it as amphibolic pathway. When we come to its rate limiting enzyme, what are its rate limiting enzyme? So rate limiting enzyme, it's citrate synthetase, isocitrate dehydrogenase, and alpha ketoglutarate. You need to remember this. ATP production is 12 ATP for 1 molecule of acetyl-CoA, and we have 2 molecule of acetyl-CoA. So the complete cycle produce 2 ATPs. And how it has 12 ATP? First, from the electron transport chain, we have 3 NADH and 1 NADH of 3 NADH. So 1 NADH concerned with 3 ATP. So 3 into 3, 9. Then we have 1 FADH2 from our ETC, and that produces 2 ATP, and the substrate level is 1. So we have 12 and the 2 molecules produce 24. Okay, so this is the thing which we need to. Its unique feature is that it's a common metabolic pathway. Here I have mentioned everything, how the step is going. So you just go through these steps, and here I've mentioned also in which step you can generate what. So that's the thing which you need to remember in this step. Guys, here in the slide, to just write it down, the raw materials. Raw material is similar, that is acetyl-CoA and our oxaloacetate gets cut. So importance of TCA cycle. What are the importance of TCA cycle? That is, it is the final common pathway for the oxidation of carbohydrates, protein, and fats. Second, it's play a major role in gluconeogenesis, transamination, deamination, and fatty acid synthesis, and it utilizes indirectly to about 2/3 of the total oxygen consumed by our body, and it generates 2/3 of the total energy. So the amount of oxygen it consumes is total amount it is giving in the form of ATP. So I have already discussed what is known as amphibolic pathway. So just read it out once over here. Then the vitamins concerning over here is thiamine. So another name for thiamine, or what we say, the thiamine is also known as vitamin B1 in the form of thiamine pyrophosphate. Then we have riboflavin, that is vitamin B12 in the form of FAD. Then we have a niacin, that is vitamin B3 in the form of NAD. And then we have a pantothenic acid as a part of coenzyme A. So you need to go through these vitamins also. When we talk about the inhibitors of TCA cycle. So the inhibitors of TCA cycle, the enzyme is aconitase, and the inhibitor is fluoroacetate, which is non-competitive. Then we have another enzyme, that is alpha ketoglutarate dehydrogenase. If you go through the cycle, you come to know that we learn this enzyme. Inhibitor is arsenic, that is non-competitive. And then we have a succinyl dehydrogenase, and its inhibitor is malonate, that is competitive. So guys, just remember the enzyme and it is inhibitors. It is very much important, and it is coming directly in your MCQs. Moving forward, now let's discuss. One more thing you need to know over here, the basic difference between a glycolysis and TCA cycle is that TCA cycle is strictly aerobic, whereas glycolysis is aerobic as well as anaerobic. Another question which might come, that we say that the TCA cycle occur in if anybody asks that is TCA cycles occur in red blood cells, so you can say that TCA cycle does not occur in red blood cell because red blood cell do not have our mitochondria, so they cannot utilize the oxygen. But TCA cycle is an aerobic process, so it is linked to the utilization of oxygen in the respiratory chain. As red blood cells cannot utilize the oxygen, so the TCA cycle cannot not takes place in the RBCs. So there is no TCA cycle in the RBCs. Just remember this thing. Now let's discuss about our gluconeogenesis. Gluconeogenesis is the synthesis of glucose or glycogen from non-carbohydrate precursors is called gluconeogenesis. Non-carbohydrate means from the amino acid proteins we are going to make our glucose. So another name is neoglucogenesis. Its substrates are glycogen, amino acid, glycerol, pyruvate, lactate, and intermediate TCA cycle, or one might come, that is propionate. Its product is glucose. Its site is liver, which is 80% or to 90%, and major site is kidney. Another compartment is mainly cytoplasm, but partially it is in mitochondria also. It is amphibolic path. It is not amphibolic. Make a correction. It is anabolic pathway. Here, just make it. It's not working. It is anabolic pathway. Then rate limiting enzyme. When we talk about rate limiting enzyme, it is pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6-phosphate, and glucose 6-phosphate. The ATP required is 6 ATP is required to make one molecule from 2 pyruvates or 2 lactates. Just remember, we need only 6 ATPs. Its sources is beta oxidation, hormonal control stimulated by glucagon. We have already seen how it is stimulated, cortisol and epinephrine, and inhibited by insulin. Then moving forward, what are the steps of gluconeogenesis? That is, first it is conversion of pyruvate to phosphoenol pyruvate, then conversion of fructose 1,6-bisphosphate to fructose 6-phosphate, and then conversion of glucose 6-phosphate to glucose. If we go through this cycle, how it goes from the TCA cycle for making the glucose, so you can come to know about all these things. Then its importance is basically maintenance of the blood glucose level by fasting, starving, and limited carbohydrate intake. Then it is the continuing of TCA cycle, as it maintains the level of TCA cycle intermediate, and it has it. We can say one more thing. It is basically the clearing the product of our metabolism, that is disposal of lactate, which is produced by muscles and RBCs, and another disposal of glycerol produced by adipose tissue. So you can mention these 2 points also. Then moving to the another part, we have discussed everything about gluconeogenesis. One question might come that why acetyl-CoA is not a substrate of gluconeogenesis, because acetyl-CoA is a substrate of maximum, but why not for gluconeogenesis? So the answer is acetyl-CoA is not a substrate because conversion of acetyl-CoA to the pyruvate is not possible, as pyruvate to acetyl-CoA decarboxylation reaction is strictly irreversible. Therefore, it is not possible. So just write this point, it might come. And it is one of the important points I have not mentioned in the slides. Then we come to our glycogenesis and glycogenolysis. What is glycogenesis? It is the synthesis of glycogen from glucose. Substrate is glucose. Sites are liver and skeletal muscles. Compartment is cytoplasm. Nature is anabolic. Rate limiting enzyme is glycogen synthesis. Hormonal regulation stimulated by insulin and inhibited by glucagon and catecholamine, that is epinephrine and non-epinephrine. And coming to its biological importance or the importance, then its importance is in gluconeogenesis, that is formation of glucose, go to glycolytic pathway in HMP shunt, and glycogenesis, that is formation of glycogen. And when we say there is a deficiency of enzyme glucose 6-phosphate, then the disease occur is von Jurgen's disease. This is in GSD we learn about this. Coming to the glycogenolysis, as we know, substrate is glycogen, product is glucose in liver and glucose 6-phosphate muscle. In muscle compartment is cytoplasm, nature is catabolic. So glycogenesis is anabolic, glycogenolysis is catabolic, rate limiting enzyme is glycogen phosphorylase. ATP, there is no ATP produced or used. Hormonal regulation stimulated by glucagon and catecholamine. See the difference. In glycogenesis, we have stimulated by insulin, whereas here it is stimulated by glucagon. It is inhibited by insulin, and whereas in glycogenolysis, it is inhibited by glucagon. So remember these 2 points. Now moving to the electron transport chain. Electron transport chain is very easy. It is another name is known as respiratory chain or cytochrome oxidase. Its substrate is NADH, FADH2, oxygen, ADP, and inorganic phosphate. Then its product is NAD plus FAD and H2O. That is the result of our oxidative phosphorylation. Compartment is an inner mitochondrial membrane. It is very important. Sites are complex 1, 3, 4, where ATPs are produced. So we say there are 3 ATPs produced. One ATP as complex 1, then second ATP as complex 3, and third ATP in the complex 4. Complexes are complex 1 NADS, that is CoQ oxidoreductase. 2 is succinate CoQ oxidoreductase. Third is CoQ site, or we say cytochrome C oxidoreductase, and 4 is cytochrome C oxidase. Components are nicotinamide nucleotides, flavoproteins, iron, sulfur proteins, coenzyme Q, that is ubiquinone, and cytochromes. Inhibitors, so you need to know all the inhibitors by their complexes. Complex 1 barbiturate drugs. So in complex 1 we have the production of ATP also. Complex 2 malanoate. Complex 3 acid antimycin A, and complex 4 cyanides. Then HMP shunt. HMP shunt is very small, and whatever the important topics which I think it might come, I have mentioned in this slide. That is, another name is pentose phosphate pathway. Then we have the phosphogluconate pathway, direct oxidative pathway. Substrate is glucose 6-phosphate. Product is ribose sugar, NADPH, and carbon dioxide. Sites are liver, adipose tissue, gonads, adrenal gland, macrophage, mammary gland, mammalian gland. In the lactating, it is very important. Mark it as an MCQ. Compartment is cytoplasm. Nature is catabolic, anabolic. We have already seen when we talk about in our first slide. Rate limiting enzyme is glucose 6-phosphate dehydrogenase, ATP. So no ATP is consumed directly or it is produced. Its importance in the production of pentose sugar, mostly the ribose sugar for nucleotide and nucleic acid synthesis. Then we have the production of NADPH. The production of NADPH importance, write it down, for our basically support for the production of superoxides and all free radicals. When we talk in our further discussion, we'll see how it is happening. It prevents hemolysis of RBCs. It detoxification of many drugs takes place, and the reductive biosynthesis of fatty acids, steroid, and cholesterol comes in the production of NADPH. Then it protects RBCs against the hemolysis. So you need to remember that if the examiner asks how it is protected, so in RBC there is no production of free radicals like superoxides or superoxides, so which cannot cause oxidative damage to the RBCs, cell membrane, or it leads to hemolysis of RBCs. So this is the reason basically. So here the steps of HMP shunt is oxidative non-reversible state, that is generation of NADPH, and non-oxidative reversible phase, that is generation of ribosome, that is precursor. Then we come to the lactic acid and The another name for lactic acid is CORI cycle. Its substrate is lactate. Its product is glucose. Its nature is anabolic. So let's discuss about its pathway quickly. That is lactate produced in the skeleton muscles. That is during when we do intense exercise and RBCs by the glycolysis and aerobic glycolysis. Then it transported to our liver and kidney through the blood. Then it reforms glucose by gluconeogenesis. Then it goes to the glucose, becomes available in the form of the circulation for oxidation in tissues and skeletal muscle. So this is the pathway which we have already studied. I have just recap it. Then its importance is CORI cycle decompose lactic acid which is produced in the muscles and the RBCs and prevent lactic acid accumulation and also prevent lactic acidosis. It provides additional energy for lactate via glucose and muscular activity. Then we talk, this is the only thing which we need to remember in the lactic acid, nothing more or extra you need to remember. Then we come to uronic acid pathway. Uronic acid pathway, its synonym is glucuronic acid pathway. What is uronic acid pathway? It is the alternative oxidative pathway for glucose for the conversion of glucuronic acid and ascorbic acid and pentose and is referred to as uronic acid pathway. So its site is liver, its substrate is glucose 6-phosphate, its product are glucuronic acid, pentose sugar as well as the one more thing that is our ascorbic acid, which is not produced in the humans. Then it's important the source of glucuronic acid, and its glucuronic acid is required for the synthesis of glucosaminoglycans and proteoglycans. Another importance which we can see that it is a deficiency in the pathway leads to the condition of essential pentoseuria. So these are the importance of it. So as I say that the ascorbic acid is not produced in the humans. Why? Because the enzyme known as the L-gluconolactonate oxidase, it is basically need to this enzyme is required to convert our L-gluconate to ascorbic acid, which is basically absent in humans. Therefore, we say that the ascorbic acid is not produced in the human body by this pathway. Then we see what are the regulation of blood glucose. If you have studied about the metabolism of glucose, glucagon and insulin in our lecture diabetic ketoacidosis, you can easily recall this, that is insulin. Insulin increases glycogenogenesis, glucose uptake and glycolysis, and it is decreased by glycogenolysis, or we then coming to its effect, that is basically its effect is insulin lowers blood glucose level immediately by enhancing glucose transporter. We see GLUT4 transporter when we discussed over there into adipose tissue and muscles. Then we see glucagon. Its action is it increased glycogenolysis and gluconeogenesis, and it increased blood glucose. Coming to another epinephrine, norepinephrine, it increases glycogenolysis and it is also contributing in our increased blood glucose. Cortisol increase gluconeogenesis, increase blood glucose level. Growth hormones increases glucose production in the liver, and it also decreases glucose uptake and insulin sensitivity. So it also increases in this fashion the blood glucose. And last is thyroxine. It increase uptake of glucose, glycolysis, gluconeogenesis, glucose absorption by GIT, and eventually it increases or contribute in the blood glucose. So this table you need to memorize by heart. So that is for all you need to learn in the carbohydrate metabolism. All the cycles which is concerning I have mentioned in a very short, so that you can go through what are these. So just you need to remember is open your textbook, open the cycle, just read out once, go through these slides, read out once and your complete concept gets clear. Just remember the first slide. You need to remember what is amphibolic, what are anabolic, what are catabolic pathways. They are coming in the direct MCQ question in the exam, so do not forget. Just go through our diabetic keto acid. You get the carbohydrate metabolism, that is insulin and glucagon metabolism pathway in a brief, so you can better get an idea what how our carbohydrate metabolism is going through. So just guys, make sure go through this PPT as well as the diabetic ketoacidosis lecture, so the entire your carbohydrate metabolism base is clear. If you like the content of my video, do like, do subscribe, do share with your friends, and put the review in the comment section if you like the content, if you like the way I'm teaching, and if you need any changes in the slide or the way of teaching, or if you're getting any complication, or you need any topic which you want me to explain through the video, just put it in the comment section and join our Telegram group for our MCQ series as well as an important mind maps mnemonics. You find it beneficial. So guys, we'll meet in another lecture with another topic that will make it easy. Till then, bye and take care.